Hi, James.
Hey, um, oh, you're using that room?
Yeah.
That's fine. If it's empty, she may be in 4004 though,
4004.
just across the small conference room
Okay.
across the hall.
No, I think she's just not here yet.
Okay. But we usually meet in that other room because
Hmm.
we didn't officially have the room you're in until
Yeah.
4, I booked that 404 conference room.
Oh, there she is. Hello. Let's start. If you want to join Zoom, that would be. James, you have to allow me.
Sorry, I'm just There you go.
And then be widening it down. Yeah, that's it.
Yeah. All right. So lecture 15. My name is Pedro Dal Castel, and this I think is going to be our last lecture. Still on compartmental cells, and there are two exercises, two reproduction exercises that me and James thought would be interesting to do today. The first one you're a little bit familiar with already. I showed last week, which is the compartmental cell migration. And the other one is the planar cell polarity, which I didn't show the model, but I showed you the images where the cells polarize to and create a large scale directionality for the tissue. And we are going to build both simulations. And if we have some time, I will also show you an extra material on pure Python programming in CompuCell and what are the advantages and disadvantages of running simulations with pure Python coding. All right. So for the planar cell polarity simulation, this is something observed in development of fruit flies, if I remember correctly, specifically in the wing, because the wing has a directionality for the cells. And biologically, one of the explanations of how these tissues achieve large scale polarity, and that means all cells are aligned to a particular direction. So how do they achieve this polarization pattern is one of the ways, one of the conceptual models is that they express certain membrane proteins that attach to different membrane proteins of the neighboring cell. But these two types of proteins segregate in the cell membrane. So they cross-inhibit each other. So this is what we refer to as heterotypic affinity. So you have two types of protein, protein A and protein B, and A attaches with B in another cell, B attaches with A in another cell, and they are expressed in different locations of the cell membrane. So with that, you can propagate the polarity of the cells. To model this different affinity between these two different proteins, we will use clusters. So each cell is a cluster of compartments, and we have 4 compartments. We have the lateral compartment, the distal compartment, the proximal compartment, and the cytoplasm. And I should have a drawing here, but I will draw this. So we have the cell here, or the full cell is there, and then we'll have a distal compartment, which is the part of the cell facing front. So this is the polarity of the cell. Then we have a proximal compartment, which will be in the rear of the cell. And then we have a lateral compartment, which will be on the sides of the cells. the cell. The central part is the cytoplasm. So, and this is our entire cell. So, four different compartments. And to achieve polarization, two things happen. Two things have to happen. In the same cluster, these compartments repel each other. But between clusters, you will have the distal connecting with the proximal of the other cell. And same thing here, the distal connecting with the proximal. And the lateral connecting with the lateral of the other cell. Lateral connecting with the lateral of the cell. If we impose these two conditions, we can segregate all compartments in all clusters, and then we achieve macroscopic polarization. So that this is the idea. Okay? Probably the only catch in this exercise is initializing the cells with all the compartments in a way that is truly random, because we don't want to predefine a polarization for the cells, right? Because if we build it like the way we were building before, like stacking compartments like this is the cytoplasm, and then this is the distal, this is the lateral, and this is the proximal, then you already know what's going to happen. The polarization will be following your predefined cell structure. But we want the cells to we want these macroscopic polarization to emerge from the cells, and the initialization has to be truly random. So that's the idea. So the initialization is the catch. I will give you like 5 to 10 minutes to figure out a way to initialize, and if you don't, then I will jump in. So feel free to start. I think this is all. This is all it's needed for building the simulation, and you already know how to handle compartments, so shouldn't be much problem. Oh, I'm muted. Okay. For now, I'm not gonna mess up with any of this. And
Is it clear? Sorry, is it clear how to do all of those? You've covered the internal contact energies before, right?
Yes. Yes. Last week.
And is it clear that what you're trying to do with the idea of the particles moving within the cells as a sort of a pseudochemical species?
Mm-hmm. Is it? Is it clear what we were trying to do?
Oh, yeah, yeah,
Okay.
definitely.
So I don't want to...
Sorry, since I can't hear her, I have to ask you I have to ask you.
So I want to do the initialization of the cells in Python. So I just deleted that. And I guess the most effective way to doing it is... Oh, wait a second. Let me revert the change. I have to set... Let me just get these to values that make sense. Okay, so each cell is going to have 100 pixels in total. So I'm going to create 10 by 10 cells from 0 to 100 in both. Just the cyto cell though. So I have a predefined square of cells, like a chessboard of cells. So if I just run this, yeah, yeah. But this is good because now I can, in each of these cells, I can create the other ones and assign them to the same cluster. So I don't need in Python to create each individual square because the squares are already there. Okay, so I'm just facilitating my life so that in Python I don't need to like scan the lattice, I can just scan the cells right away. Okay, so that's the idea. All right. So here in Python, the strategy, I think that the most effective strategy, let me go to Paint, is to, in each of these squares, we randomly paint pixels of each compartment. We scatter these pixels around the cell. That's the idea. So all the cells will be randomly arranged, and then these compartments will segregate inside the cell in any way, okay, in their own way.
So like when the simulation starts and they will just combine, they will like self-organize.
Yeah.
But like it's not even like a cell because like for example like this part it only has this red part compartment, but it doesn't have like the green compartment. But like if you have a list of cells and even though like they didn't have a very polarized like arrangement, but each cell would still have like the red compartment and the green compartment.
So I don't know if what I said was clear. Let me try again. So we each square of these is one cluster. Okay?
Oh, okay.
So this is each square of this is one of those squares of the grid. Okay? And then within each of these cells, we randomly scatter pixels of lateral, distal, and proximal compartments.
I'm sorry, I thought like you're going to like just split this big square, but now I understand.
Right. So I'll just go ahead and try to code it myself. So we start with a simple for loop. So for cell in self dot cell list by type and then self cyto. So here for each cell, we want to take the pixels. We want to scan the pixels of this cell. And I don't think I have the pixel tracker plugin, so I'm going to back to the XML and load the plugin somewhere. Pixel tracker. Now back here, I want to scan all the pixels of the cell. So visit pixels, cell pixels. Okay. Nice. So I have the cell. So I'm scanning each of the cells and I'm getting the pixels. Okay. Now I want to create before I scan the pixels, I want to create the lateral, the proximal, and the distal cells or compartments of this cluster and assign them to the same cluster ID of my cyto compartment. Okay. So to do that, I create a cell. So cell manipulation create cell. I just want this part. And I will say one for the proximal. So this will be the proximal cell. And then one for the lateral and one for the distal. This, so I have the 3 cells. And now I need to assign them to the same cluster ID of this one. So cluster ID reassignment. And I also want the cluster ID. This one comes here. And then I have to repeat this for each of my cells. Okay. And I prefer to have this organized like this. So I have the creation of the cell and the reassignment of a single cell. So this is all one cell. This is another one. This is another one. Now we want to insert these cells inside the cyto. So cyto again is this guy here. And this one is this one. So proximal, lateral, and distal. Lateral, proximal. Okay. Cluster ID, cell cluster ID. We could even call this one cyto just for standardizing the syntax. So this is cyto, 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 proximal, 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 lat, 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 this to this to this. Okay. Now, when we scan the pixels of the cell of the cyto, we want to rewrite some of them randomly by pixels of prox, lat, and dist. Okay. We already have NumPy, so we can say, let me grab the cell creation syntax again. So create cell detailed. I want this cell field equals cell. And at a position X and Y. And I want this, and if I'm not mistaken, is dot pixel dot x and dot pixel dot y to get the coordinates. I don't know if there is if I do this cell pixel, how does it do? This one is pixel tracker dot pixel. Okay, yeah. So it's dot pixel dot x, then dot pixel dot I to get the coordinate of one of these pixels. So now I want to overwrite this pixel by something with a certain probability. So for example, 25% chance I overwrite with prox, 25% chance I overwrite with lat, and 25% chance I overwrite with dist. And 25% I keep it as Saito. Okay, so that's the idea. So here I will do np dot random dot choice, and I have 4 choices for this guy: prox, lat, dist, and Saito. And this should be it for the creation of the cell. So I'm going to run this and see what happens. Oh, it worked. It worked very nicely. So if I zoom in, this is one cell, and then the gray is Saito, the red is dist, the green is prox, and the purple is lat. Okay, so I have all the cells here in the same proportion, so 25% each, and the same thing for all the cells. So this is a very simple way of randomly disposing compartments within clusters, and it is completely random, pseudo-random. The tissue can polarize in any given direction. We don't know which direction the tissue will polarize to. So this is a good initialization strategy. Okay? So I'll keep showing, well, okay. I'll keep showing you this in your Zoom, and then you can work on it. Try to get the same.
I think actually like our like thinking way is the same because I also wanted to.
Yeah, but
using
the
Excel
problem is you have to reassign cluster ID later.
a lot of time. Yeah, and I have to like that's right. Okay. I can show like the cells. Like amazing stuff.
Show me your XML. Oh, no. This one is the minimum, this one is the maximum, so it's 1 to 200 right there.
Oh.
Yeah. Yeah, that's it.
Thank you. Yeah, it's just a little typo here.
Mm-hmm. Yeah, there you go.
Okay.
Now, your objective is to get this to segregate. So if I run this, it's not exactly what we expect, right? It's not exactly that beautiful yeah.
Because you didn't change the
Yeah.
concept.
And there is also another thing here. We should have 2 lateral compartments per cell, right? So there is a small change I have to do here. We have to create a second cell, a second lateral cell.
Two.
Yeah, lateral number 2. And also, and so having two compartments, basically. Yeah. And probably I have to increase the size of the cells to reflect that. So I'm going to do with 20. So the cells are 200 by 200. And then I will do lateral will be 25, 25. This will be 50, 50, and this will be 75. And then if I get the contact energies right,
This is um this is contact energy between cells. Like
yes. Mm-hmm. And then I guess I have to do
should increase the contact energy between lateral 1 and lateral 2.
yes. So this one is 20. And then proximal with distal 20. And then everything else, yeah. I think that should be enough. Let's see what happens. Nope.
Remember that the contact energies add. So you may want to go back to the contact energy list that Julio specified.
Ah, so if I look at this cell here,
So,
Saito is in the center. Then we have, oh, I think I know what I have to do. Now I'll set all of these to 0 first. And just see what happens. Okay, this is good. Yeah, this is very good. Okay, so now they have some structure. And distal with proximal tends to connect, which is good too. Now I can try to force proximal and distal to separate and lateral with lateral to separate. Let's see what happens. Okay, what is the green? Proximal. Okay. Proximal is
tend to like stay at the center of the cell.
yeah.
It doesn't separate with the cytoplasm.
Yeah. We have, okay, so we have lateral connecting with lateral. You can see lateral is on the outside of all cells connecting. And is this, I don't think this is periodic boundary condition, by the way. It should be.
But then choose that would be like no flux.
Yeah, but it has to be periodic. I don't want the boundary playing an annoying role. Okay. What would you suggest to do now?
I think like so firstly you can see like there are like two distal parts in
Mhm.
one cell.
Mm-hmm.
So like for each compartment, they're not very integral or complete inside of one cell. So you have to like
Cohesive.
insert the other one.
Yeah, I can also reduce their size to 25. Maybe that's going to make things look better, more similar to the images. Okay, so we don't have cohesion yet. So maybe we would like the cytoplasm to be more cohesive and the other things to behave more like membrane. So to do that, what would you suggest? Yeah. Do you have any idea?
Um, it's just like increase or level up the contact energy of Saito with other
Mm-hmm.
compartments a little bit, but
Mhm.
Saito and Saito still like
This one doesn't matter.
yeah, you want to kind of like
This one doesn't matter, right? Because we just have one Saito compartment in the same class. Yeah. Okay, so now we have our Saito is more well behaved,
Mhm.
right?
Mhm.
And so we have lateral connecting with lateral on south. We have distal connecting with proximal for the most part. So you have green with red, green with red, green with red, green with red. What else? Maybe for now it's just a matter of size. So let's what I can try is increasing the size of Saito a ton.
Shrink the size of the lateral.
The other ones,
Yeah, because sometimes you could notice like the lateral is tend to wrap up the whole cell.
yeah, true.
So we just introduce some new ones.
Yeah. And another thing I can do is see how in some cells we have um um
Like the
hmm,
components is below.
this one is curious. Let's look at this one.
Mhm.
We have some lateral here, a lot of lateral here. Of course, there are two cells, so if I visualize cell borders, okay, it's split here, so there is one lateral cell here, another one here. Oh, these two should hate each other inside the same cell, right? And maybe they don't. Lateral with lateral. Let me put this to 100. Lateral with lateral should hate each other. Distal and proximal, too. They should hate each other. Let's see what happens when I do that.
Oh, cell bond? Or maybe it's like too strict so that so originally in one cell like the pixel of like some of the pixels belong to the distal and there are other pixels that belong to both the distal compartment and they're separate. So if you like and there is like a probably proximal pixels in between. And if you like set the threshold too high, like those two parts of distal pixels cannot compensate. Like they cannot meet each other because like there's a very high energy threshold between.
Mm. So when they get stuck.
Yeah, so you can see like there are more separate
Mm-hmm.
proximal and
Mm-hmm.
distal pixels in one
Mhm.
cell.
Mm-hmm. Mm.
And I think it would be good if you set like two lateral cells. Because if you like expect
Yeah, I do have two lateral cells. For two lateral compartments per cluster.
compartment,
Yeah.
but you can't like mm, uh, but they're like with the same property because they're still like the same cell type, but like one cell type was split to two compartments. Okay.
Uh, what do you mean?
Mm. Like if you like have two lateral cell types, it could be helpful to be honest.
Ah, mm,
Because now you only have like one lateral cell type, and
yeah,
you expect it to be like, so one is at the top and one is at the bottom, or one is
one side, the other one's the other side. Yeah.
yeah.
Yeah, but then we are changing the model structure. Let me pause this and look at it. So if we take this cell here, it has proximal with distal
Mhm.
and distal with proximal on the bottom here. There is some connection proximal with this other cell.
Mhm.
So maybe should try to kill this
Mhm.
and kill these all the cross connections that are not distal proximal between clusters. So in this one, I should have, for example, distal 100. I should have this one 5 is fine. This one could be 100. This one 100. This one 100. Let's see what happens if I do this. They should really hate each other. There are some things getting stuck. Yeah, like they separate. So there is one piece of
Oh,
one piece here, one piece here, one piece here.
because like
Yeah.
it really hates that, so you couldn't cross over this
Yeah,
part and
yeah, they get stuck. They get stuck, right?
Mhm.
Um,
Because you saw that as like 100. So
yeah, yeah. So are you suggesting that maybe if I
like like just decrease it a little
Mhm.
bit.
And maybe also decrease their volume. I feel like their volume should be they should be way smaller than this. They're too big. Maybe because they are too big, they can afford to have different clumps everywhere. So maybe I could do like large volume here, like this, and then have them 10 pixels, 10 by 10 pixels. Oh, but wait a second. I set this to 20. So each cell is 20 by 20 is a 400. So I can, this guy can be 240, and then I can do 20, 20, 20. Let's see what happens when I do this before I change anything else. Okay, that's much better. Maybe. Um,
So like, oh,
yeah, we need more mixing now. They're very stuck. So maybe we just need more mixing with cytoplasm. Something like this one, let's go down to 1 here. So there is some segregation, but they can mix if they want. So they can rearrange internally inside the cell. Let's see what happens when we do this.
couple of seconds.
Let's do 0. We can even go negative. I'm not sure if he does this, but yeah, so you can see that there is some mixing.
Mhm.
So the excess is trapped inside the cell.
So, like for example, like there is some like mixing here.
Mhm.
So probably it expects this cell have a proximal protein at this side, but doesn't because you see the proximal
Mhm.
contain most space. So that not just go this side combine it with this. So you just like
Yeah, because
mixing.
they're too stuck. One thing we can do is relax this one a little bit. Like let's do 20. So relax the cytoplasm with everything else. So maybe we want something in between those two. But this one is already much better than what we had before, because now we can see some polarity. Now like we have red, green, red, green, red, green. Here, look at the bottom one. We have green, red, green, red, green, red, green, red, green, red. Okay. Yeah, so this is already looking much better. The one problem I have with this is because it's not a hexagonal grid, we can have cells in each row with a different polarization. So for example, the bottom row, we have red, green, red, green, and the row above we have green, red, green, red. So maybe we really want this to be a hexagonal configuration and not a square initialization.
Why would like configuration like this would help this to align in both?
Because then the diagonal cells will interact. Sorry, in a square configuration, the diagonal cells don't interact much. That's the problem. We want to have them interact so that the red here repels the red there. Right? So we want these two guys here and these two guys here to repel each other. And when the cells are in a square configuration like this, they don't come in contact. Okay? So we would love to have a hexagonal configuration first, to start with. So one way of achieving it is to, instead of creating these clusters in the start function, we can do with a pif initializer. We can run it like this until it achieves a what is cytos? Okay, good.
Actually, this is how you like to form a hexagonal configuration. It's still like using a square lattice, but
Yeah.
hexagonal cells.
Yeah.
Okay.
We can also go into a hexagonal lattice and see if it helps. What happened? Did you understand what happened?
No, I thought you like clicked somewhere.
No, I didn't do anything.
Run it again.
Yeah, because like you
Your target volume must be small, and there must be one white voxel somewhere, but not from there, as if it should.
like a periodic function.
Well, I'm going to say this is pretty much hexagonal. Is this hexagonal to you?
Yeah.
Okay. So I'm going to come here and say generate pif. And then the rest you already know. So this is going to be instead of this guy, wait, instead of this guy, we will have a pif initializer. So steppables, pif initializer. And then if we run this, perfect. Okay. Now I reverted the change here. So it's going to start from a hexagonal configuration and then it creates the cells like that. And now because it's in a hexagonal configuration, the diagonal cells have to interact.
Mm.
And maybe now it's going to achieve something, maybe. Let's see. Might take a while. Lateral is connecting with lateral. Here we have green, red up here.
Mm.
Here we have green, red, green, red, green, red. These two here are too close to each other though. Here we have green, red, green, red.
Lateral combined with each other in boundary.
Now we can have cytoplasm hate cytoplasm more. I think I decreased that to get the hexagonal configuration, but we can put that back to 20.
I think that there are some cells usually, I think like they're never going to stay
Yeah,
stabilized here.
yeah, I agree.
It's like this cell you couldn't detect the red
Yes,
part of that
yes.
cell.
That's never going to change, yeah.
So it's like you want the cell itself.
Yes. And also like this one, we have the red neighbor to the green. And
You know, they're
so,
too close.
yeah.
they're not-
Yes, exactly, exactly.
But if you have a example configuration, that could be a problem because you have 6 at this one.
Mm-hmm.
So like if the pixel goes to adhere to, for example, like this edge,
Mm-hmm.
and it could, so you expect the proximal, for example, this is the edge, and you hope the proximal
Mm-hmm.
compartment to go like to the opposite direction,
Mm-hmm.
that edge, but in fact it could probably just appear to the neighboring.
Mm-hmm.
So it has like more choices now.
Yeah, there is one other thing I can try, which is increase the mixing between lateral and everything. So They're already mixing very well.
I think it's still like you should have two lateral parts.
Two lateral types, but then we're changing the core model. Maybe just have to relax things a little bit. Let me try 20 on everything. And let things relax more. Okay, this is looking much better.
Yeah,
Yeah, this is looking way better.
because you can observe like those unstable
Yeah.
cells.
So they have a better chance of finding their partners now. But let's see. This one, for example, we still have proximal and distal being neighbors. They are not touching each other because they hate each other, but they are still neighbors. And there is a little bit of proximal here separating the two. So basically what happens is that eventually this guy here has to lose to this one. And then it's transferring material from up here to down here in order to break this neighborhood here. Maybe this will happen like given enough time, like we'll have to wait a long time for this to happen.
Yeah.
And everything is interacting with everything, like all these interactions are being transmitted across cells. So maybe it just takes a long time because they are fighting each other, right? Like this cell has 3 proximal pieces and 2 distal pieces, right? They are getting stuck on the configuration of the neighboring cell.
I think like probably this. You see like this small part disappeared.
This small part disappeared?
Yeah, like originally there is a small part and you
Oh.
wish this part to come.
Oh, and it
here,
disappeared, yeah. Did
but
it
it was
lost?
like no, I think it just migrates to there
Mhm.
because there is a red. So the possibility for this part to travel to this is bigger than that one traveling.
Yeah. So we can increase even more the traveling probability of these proteins by giving negative energy with cyto. So we can do like -1. Usually -1 is enough. Like a very small quantity is enough to have a lot of mixing in CompuCell. Don't ask me why. Yeah, you see, like we can even exaggerate more. So maybe now because we have more stuff traveling inside the cell, they have a better chance of finding their appropriate partner now. And then once they find them, they stick and stay connected, right? The other thing is having smaller cells and more cells could help. Because there is less space, less traveling distance inside the cell. But let's see, is there a polarity here at all? Let's see. We have green, red, red, green. Huh. Interesting.
Probably would say too, like active.
Yeah. Yeah, there is a limitation here, which is the repulsion between these compartments are only short ranged. They're not long range, so we cannot once there is a little bit of spacing, like in this cell here, between the proximal and the distal, it's going to stay here, and they will be neighbors forever. We cannot, they cannot repel until the distal gets to the top or until the proximal gets to the side. So they are basically stuck, I believe. They are stuck there on this configuration. Unless we make the cells much smaller than this, I don't think we can achieve the desired outcome
Because
with big cells.
we want to try to like smaller
With smaller cells. Let's try with smaller cells. So I have to remove this one. Okay. This one back. So cells will be 10 by 10. And then I will have the volume here will be something like 70, and then 10, 10, 10. So they're very small. 10. This is fine. What else? Oh yeah, I want to not have this for now, so I can save the pif. And relax the contact energy between cytocells so that they get to a hexagonal configuration. And this should be enough. Now I come back here, activate the pif initializer, and activate this, and run again. Now I have a feeling I need to increase the size of the compartments, because there is too much cyto stuff. So something like 60. Let's do 50. And then have these ones have to be like 13. Is this correct? No, 16. Right?
on the vowels, okay. Oh, that one looks okay. Like we could wrap up the same process.
Yeah. Yeah, but we definitely have some polarization going on now. Because, for example, if you follow a line, like this is red, green, red, green, red, green, red, green, right? And then this cell is undecided. This specific one here is kind of undecided still. But let's see this one here, for example. We have green, red, green, red, green, red, green, red, green, red, green, red. So they are trying to polarize. So let's plot the polarization field of the vector field of polarization.
Okay.
Do you want to do that?
Oh, I don't know how to do that actually.
Okay, I can show you.
Vector, vector field.
Yeah, it's a vector field. Yeah, so to
Vector.
yeah. I mean, you can do that in MATLAB, but we want to see it as the simulation runs, right?
Oh,
Yes. So we want to create a vector field. So we come, I think it's in init that we have to do extra fields, cell level, vector field, cell level. Create. Okay. This one will be polarity. And then in the step function, we need to have something. So extra fields. Vector field, cell level. Example. Let's see the example. I don't want random stuff. Field equals, well, okay, so our field polarity will be that guy. Then we do field clear. And this one.
also like field bracket, so do you need to like change it to polarity as well?
Yeah, but I'm going to delete all of this.
Oh, okay.
Yes. I would have to. So now, what defines the polarity? It's basically the position, the difference in position, or the difference in center of mass between the proximal and the distal, right?
Mm.
So that's one way of defining it. So to do that, we scan clusters. So we can do, instead of for cell, we do for cluster. So visit all clusters. And then we visit all cells within a cluster. So visit cells in cluster. There is a typo here in the snippet. There is still, I think we found this typo, James, years ago, and it's still there. Now we have cluster, get cluster cell cluster for compartment. No, what? For cell, for cluster in self cluster, and then for cell compartment in cluster, and then I don't need any of this. Now, if the compartment type is dist, I have the dist x and the dist y being this guy dot x com, this guy dot y com, and it's better to just have this position and then something like this, like a proper vector, right? And then, so this is if the cell is dist. If the cell is prox, then we have prox position, and then we have our polarization vector will be distal minus proximal, and then this goes there. Does this make sense?
Mm.
Of course, this is per cluster, so outside this one. Looks good. And we just do this once every, no, once every 50.
So like in one cell, how many vectors would you have?
One.
One
One per cell.
cell compartments. For example, like for distal cluster, there are like multiple cells. You're iterating each cell inside it. So for each cell in the distal cluster, you would have.
No, I iterate over the compartments to get to the position of the distal
Oh,
and
so-
the position of the proximal.
Oh, so this is the compartment, not cell.
Yes,
Okay.
yes.
Okay.
This is the
Sorry, I was
compartment
kind of
within the cluster.
confused for a
And then
second.
for each cluster, we have one single polarity.
Oh, okay.
Okay.
Mhm.
I don't know if this will work, so I mean, just have to run and see. Expected something.
If
Where's that? If
yeah, you
Oh,
could
here.
have it. Okay.
unsupported for list. So let's do
You have to like
for oh come on.
this is the
Element wise difference lists. Element wise difference of the lists. Oh, okay. So I have to transform them to an NumPy array. Very annoying. Okay, let's see if this works now. Cell is not defined. Oh yeah. For cluster. Okay, there is something else I would have to do. Yeah, cell doesn't exist. So we can do like cyto and then say if compartment, then we say cell equals this, and then it associates with the cytoplasm. Hmm?
Oh, okay. Polarities. So the cells
This cell after the loop, this is identifying the cytoplasm of the cluster. And then when you do polarity cell, it's going to draw the vector in the cytoplasm.
Okay.
That's the idea. Array dimension, oh yeah. Okay, it's running. So now we can see the polarity vectors. Sometimes they go red for some reason.
Well, you see, like some of the arrows is
They are, and they are, yeah, that's unfortunate, but there is no way we can change that. The problem here is they are never diagonal. They are always vertical or horizontal, and this is annoying me much. So there is something weird going on somewhere. Because they shouldn't be.
Diagonal. Instance.
Yeah, but they should smoothly, you know, change direction, not jump between horizontal and vertical all the time like this. Because like if we look at the slide again, this is how it looks. So they have smooth directions. But this could be a problem in version 4.8 as well, so I don't know. We can try to take a look here. Fields. Scale, we can overlay. No, we can fix arrow color. Yeah, let's fix arrow color, of course. Length 1. Oh my God. Okay, so let's not scale. Okay, so let's scale and go back to 1.
Does it mean that if the vector isn't long enough, then just ignore them? Like, but that shouldn't be like the reason, because there is going to be some more stable.
No, but there is always a vector everywhere. Now, why it's not? Let's not fix arrow color then. Oh, so it's that arrow color. If I check that arrow color thing, it messes up everything. Interesting. Let's fix arrow length. Ah, let's do 5. So I shouldn't check any of these. Ah,
They're always like lower down, so vertical.
yeah. Which is very weird because like if so prox dist minus prox well this let's print this Paul thing, because maybe there is another problem going on. So
I think apparently like
6 and 2 and then -1 6. I mean they are all, you know, to all directions. Yeah, they are not either vertical or horizontal. They are to all possible directions imaginable.
issue.
Yeah, it's a problem with the yeah. So let me open a previous version of Compartmental.
Is you're using
4.8. Yeah, the latest. That was the latest. So now let's do with one of the previous. Yeah, this is 4.4. Same problem. So there might be a problem with my simulation somehow. One last try is to transform from a NumPy array back to a Python list, because maybe it doesn't like NumPy arrays much. Oh yeah, so it doesn't like NumPy arrays. Interesting, interesting. And this is, oh no. This is the 4.8. Okay. So it doesn't like NumPy arrays.
So you better document that and send it on to Matcha. Screenshot it and let him know.
Okay. And is there alignment? I mean, now we can actually see the alignment. Like these cells are aligned. These are more or less aligned.
locally.
Yeah, locally they align more or less.
You can see some like persistence.
But they're really stuck. They're really stuck here. So I successfully failed at accomplishing the task here. I mean, it has potential though, and I'll encourage you to achieve the result. There is one caveat though. I think Julio freezes the cells. So it's always hexagonal. There is no, like, the cells are not diffusing, they are fixed in a hexagonal grid. So
Only from the cytone also.
no, I don't know how he does that. Probably I don't know. I don't know, I'll have to look into his specific thing. But there is a lot of mixing between these compartments. So maybe if we increase mixing between one last attempt, I'm going to increase mixing between lateral internally and between lateral and the other two compartments and see if this helps. So that's my last attempt. No success. Overlay. Let's see what happens when I overlay. Nothing. Okay, 10 minutes break, and then we'll be back with the cell migration, the collective cell migration. Do you want me to send this to you? Yeah, to write everything. So let's get back to it. So next topic is on collective behavior. There are multiple situations where we see collective migration. For example, in birds, herds, people, fish, and they look beautiful. And cells also do it. This is neurocrest cells migrating. This is the primordium migrating laterally. This is dictyostelium discoideum, where the cells converge and create like a stalk thing. Like they do this, and then they start growing like a plant. It's very, very interesting. Cancer invasion sometimes involves collective migration, the cells collectively spreading, and wound healing as well. So we would like to have a model of cell migration that can do all these things. There are many ways of implementing migration in CompuCell, as you know. So for example, I mean, we are not there yet. So to have a model, a working model of cell migration in any any framework, either analytical or computational, you need first your cells need to move. Okay, so active motility. They need to be cohesive. So like these cells, they tend to stay together. They don't like tend to separate and move on their own. They tend to stay cohesive somehow, like either just close to each other or connected. In the case of cells, because they don't see each other, they should connect. But for example, in other flocking cases like birds, they can see each other, so they don't need to have direct contact. And also to have the kind of flocking behavior we see, they need some interaction between each other, between neighbors, so that their velocity align. In biology, active motility emerges from cell polarization. So the cell orients its cytoskeleton to a certain direction and starts polymerizing actin in a certain specific direction. And that this actin polymerization pushes the membrane forward and attaches the cell to the substrate and pushes the membrane forward. So it's, I like to use the analogy of like, imagine you are inside a big plastic bag. You are completely inside a big plastic bag. So you step on the plastic bag, you adhere to the floor through the plastic bag, and then you push the plastic bag forward. That's more or less what actin is doing here. It is inside a plastic bag. It is attaching to the floor, to the in this case here the table, and then pushing the membrane forward like this. Okay. So this is what each actin fiber or this actin mesh is doing. But there are other ways of cell migration. Okay, this is one of them. Or this is the lamellipodium type of migration. Then cohesion comes from adhesion in the case of cells. Cells will adhere to each other and connect. And also they can attract each other from distance using chemical fields. So in the case of dictyostelium discoideum, for example, each cell is secreting a chemoattractant so that even though they are scattered, they can converge. Okay. In the case of wound healing, they are all connected, and then they can push each other.
I think I've tried to similar this before, but
Mhm.
that one was with the persistence love, or something like that. So it's
Mhm.
yeah, go ahead.
Okay. Then you need somehow transmit information between cells so that their velocities align. In the case of in biology, this can happen through mechanotransduction. So, for example, in areas where one cell attaches to the other, you have like a signal transduction from the migrating cell to the back cell, and then the back cell starts polymerizing, acting in that direction as well. So there are several ways that migration can propagate to neighboring cells from one migrating cell. So people discuss like signals and also I mean chemical signals, like when something binds, this binding cascades some reaction, but they also discuss, for example, mechanical signals. Like if there is a cell pushing another cell, this cell changes behavior and starts moving as well. So there are many ways to induce polarization across cells and force them to align. Oh yeah, this is a movie. Yeah, cool movie. This is this is a molecular dynamics model. So something like Physicel, like a point based model. Yeah, it's unfortunate it isn't smooth on your end, but the cells, they see each other. And then there is a torque applied to the velocity vector of each cell to force neighboring cells to align their velocity. So it's like brute forcing alignment in this case. But in CompuCell, we want the alignment to emerge from the interaction between the cells, from the contact interaction between the cells. In this one, you just force it. You just say there is a torque, they see each other, and then they align. That's it. And then you can control how much you want them to align. And if you want them to align very well, they will flock very cohesively. And as you decrease this alignment parameter, you get less and less flocking. Oh yeah. And there are multiple ways you can implement this in CompuCell. This model here you already saw. This is what we call a Boyd's-like. So in each cell, there is a vector, a polarity vector, and you apply a force to the cell on that direction. Then you can fluctuate the angle of the polarity vector, like randomly fluctuate this polarity vector. So the cells will have persistent migration. Then you introduce another mechanism to the cell migration, which is you correct the angle of migration based on the displacement of the cell. So if the cell is moving to a particular direction, the polarity tends to correct to the direction that the cell moved. And what this does is, if two cells are moving to this direction, for example, and they collide, they cannot go past each other. They will keep moving to this direction, and then the polarity vectors start aligning because they are pushing each other and moving in the same direction. So the displacement feeds into the new polarity vector, and then these colliding cells will tend to align. Okay. These creates this simulation, which I think you already saw running. There is that simulation where you can change the number of cells, the adhesion parameter. I can show that to you. I don't know if I sent the simulation to you, probably not, but it's where is it? This one. This is the simulation in the GIF that I used to generate those GIFs. So this is the simulation, and you can change like the number of cells. I think we showed you this already, no? No? Okay.
I think I've seen this once, but
Mhm.
it's the first time that I saw you using the steering.
Yeah.
Yeah, I've seen that once, but like not very deep into the code here.
Mhm.
Something else.
Yeah, I can show you how the code works, but this is very complicated to generate. There's a lot of maths with angles and stuff. The one we are going to do today is much simpler than this
Okay.
in terms of implementing it, in terms of coding. Right? And then with that simulation, you can study, for example, the number of clusters. And here I am changing the surface tension between the cells, the affinity between the cells, how much they like each other. And you have a larger number of clusters as they hate each other, as they increase the contact energy between them. Okay, makes sense? So if they love each other, you have a very low number of clusters, very, very few clusters. And as they hate each other, you have more and more clusters. There are 1,000 cells in the simulation, so this number tends to 1,000. If they absolutely hate each other, they never get in contact with each other, so each cell is a cluster. You can also define a collectivity parameter. So there are these beautiful mathematical formulas here for getting something that captures the collective behavior of the system. So phi is the mean alignment of cell polarity. F is the polarity vector. So you're getting the average of that. So the more the cells are aligned, the more phi is close to 1. And then you can also get the rotational alignment. Because if two cells are doing this all the time, if they are always like orthogonal opposite to each other, their speeds, they rotate. And this is also a form of collective behavior. So they are misaligned, but if they are swirling like this, they actually have collectivity. So there is one metric for parallel alignment and another metric for swirling alignment, like a circular a vortex kind of behavior. And then you sum the squares to get like a final metric that considers both. And then you can plot this collectivity parameter versus something like the persistence of the cells. So here, as you increase persistence, you have more and more collectivity. And as you decrease persistence, you have less and less collectivity. And these movies show how they look like. So this point corresponds to this simulation where the cells are very persistent and then they tend to migrate to the same direction for longer periods of time. And this other point here corresponds to this movie where you see the cells are constantly changing direction and then they cannot establish a collective migration polarization. These curves change as you change the density. And yeah, interesting. So now there is this other way of getting collective behavior, which is using the self-generated gradient model. I think you did implement this in the chemotaxis lectures,
Yeah.
right?
Yeah.
So you have a cell, it depletes nutrient, and then it chemotaxis towards the gradient of the nutrient field.
Yeah.
And these are ways of calculating velocity, like a measurement of velocity for CompuCell cells. And then you can plot something, for example, when the cells are fastest, or like the speed of the cells versus the nutrient recovery time. If the nutrient recovery time is very low, very fast, then the cells don't have to move much. If it takes too long to regenerate, then the whole field decreases, and then the chemotaxis is less efficient. And so there is an optimal recovery time where there is a trail behind the cells, so they self-avoid. They avoid their own trail. But it's recovering fast enough so that the cells can always find a path with nutrients. And finally, the one we are going to implement today is a self-polarizing cell. So here we don't impose any polarity for the cell. And we also don't impose any alignment between the cells. So both things emerge from the interaction between cells and how we set it up. So we build clusters, 2 compartments. We have a front and a back compartment. Then we have an internal field in the front compartment that protrudes. It does chemotaxis towards the medium. So it pulls the back compartment. The front and the back compartment has some affinity so that they don't completely separate. But we also want them to segregate internally.
Oh, okay.
Yeah. And finally, we can also define the contact energies between neighboring cells so that they align collectively. And in the first slide, I have these videos of different types of migration you can achieve with this. So you can have them migrate side by side. You can have them do swirls like this. And you can have them migrate in a line, in a queue like this. So you, from the same model, just by adjusting parameters, you can achieve various kinds of collective migration, which is very interesting. And you are not forcing alignment. You are not imposing, for example, oh, if there is a velocity vector in this cell and in this cell, I'm going to align them. No, they emerge from the interaction, the contact interactions. So to reproduce this simulation, we need 2 things that you're not familiar with. Well, the compartments you're already familiar with. So you need to have compartments for this. You don't need links,
Okay.
but you can have links to increase cohesion if you need. Also, you can have a nucleus, but you don't need to. I usually don't like putting a nucleus in there because it makes the simulation, the time scale of the simulation increase drastically, because the nucleus is like damping migration, slowing down the cell migration in
Why?
CompuCell.
Why?
In CompuCell. Because it's like it's another cell that you have to drag in the
Right.
simulation.
I'm going to intervene there and say, though, the nucleus does have different mechanical properties because it's rigid. So if you want to know how cells crawl through channels, for example, it makes a difference. So to start with, you definitely don't need the nucleus. But if you're actually going to do biomechanical simulations and you want to understand cell alignment, co-alignment, then having the nucleus can be very interesting.
Yeah. And the two compartments that you absolutely need, so the starting pack is a front and a back compartment. Those are necessary. Then you need an internal field in the front compartment. So I'm using the word lamellipodium here, but this stands for the front compartment. The lamellipodium is the actin mesh that is in the front of the cell. So this internal field is always 1 in the front compartment and decays very fast everywhere else. And then you have chemotaxis of the front compartment towards medium based on this internal field. And also towards the cytoplasm of other cells, the back compartment. Maybe I should, oh sorry, maybe I should just change those words quickly here. So towards medium, this will be front, inside the front, and towards the back of other cells. And then we have to, in the chemotaxis plugin, we have to disallow chemotaxis between compartments inside the same cluster. Otherwise, the front will also have chemotaxis toward self back. Okay, and you don't want that. So there is this, oh, it's not here. What? Wait a second. Let me open that simulation. There's somehow that piece of code went missing. I think it's this one.
Let's see.
Yeah, it's this thing here. Disallow chemotaxis between compartments. Yeah, this is the option that you need to have cells like the front compartment migrate towards the back compartment of the neighboring cell.
You don't- So,
Yes, but you don't want itself to crawl towards the back of the own. It's the self cluster.
what's the wow,
Yeah, this is like the back because I call it myo for myosin in that simulation. But I think using the words front and back makes more sense. Okay?
Mhm.
Do you think you can generate the simulation with this information here?
I'll control.
And I can supervise you as you go.
Mm.
And you can also start with the cluster initialization that we used for the tissue folding. That one is very good for this kind of exercise.
That one, okay. Mm.
Yeah, you don't need any of this. This is for the links, right?
This one? Yeah.
Type. Yeah, this is for
Yeah,
links. Yeah, you don't need that.
I'm thinking about like reselling IP.
Front for sound service type of
things.
Yeah, you don't need the basal part. Yeah,
So that could just assign the plus the
Yeah.
go to the cell.
yeah, that's it.
Because I copied the project here
Yeah,
and made some notifications. So what is that doing? I have to change all. don't have any idea what it was crash. I just like try to initialize first. I like to roll 6 cells.
What, it just crashes?
Yeah. So if you try to open the folder, right.
Yeah, it's usually not a problem in Python because when it's a problem in Python, it tells you what it is. It's usually a problem in the XML. Something like the brackets, for example. Mm,
Can you
Oh.
screen share?
there is a problem. You have chemotaxis of a field that you haven't specified anywhere. You have to create the field.
Function FE.
Yeah. Steples.
Stepable?
Yeah, it's a steples.
This one.
Yeah.
Okay. Uh-huh.
Okay, I can copy and paste the part. Oh. And then it has to end with this other line. Yeah, I sent you the code. And I didn't send you the planar cell polarity thing yet. I'll also paste on the chat for you.
Could this stepable be inserted between
Yeah.
line? Okay.
Yes. Just make sure the name of the field is the same. Yeah, so substrate.
That's proposed.
No, doesn't need to be that fast.
Oh,
Substrate, energy, yeah. And then the cell types have to be the, yeah, this is back. And then back, and then front. And then front. Chemotaxis, okay, so here you have in chemotaxis, you have chemotaxis towards back, but you also want chemotaxis towards medium.
Oh, okay.
So, yeah, duplicate. Yeah,
Just delete
and
it.
it's a separate, it's a separate chunk.
Oh, okay.
Yeah, so undo. And then copy the whole thing, yeah. There we go. Now, the chemotax towards medium, you don't need the disallow.
Mm-hmm.
That's actually not going to work if you do that.
Mm-hmm.
Yeah. And then the lambda values have to be negative. Because the field is high inside and low outside.
Ah, okay. Oh, please, what the two?
Negative here too.
And it says. Oh, okay.
Yeah.
Mm.
Yeah, try.
So it's
A mismatch. Did you did you paste the other line, like go to the field part?
Oh, I think I missed some stuff.
There is a, there we go, yeah. There is a
Yeah,
missing,
you have another. Yeah, here.
and that's the planar cell polarity code for you to play with.
So it
Nice.
looks like that. Yeah, I think.
Yeah.
Oh, it wouldn't.
Could you screen share it, Pedro, so I can see?
Um, I'll let, shall we share? Shall we?
Oh,
I don't know how
did
to,
that. Yeah,
I'm not gonna try.
it's definitely
Hmm?
it's definitely correct. Okay, so this is how the initialized flaw looks. So
You're not sharing your full screen.
really?
Yeah, you're just sharing to edit.
Okay. Oh, nice. Just...
Go stop share.
Okay, there we go. I can see it now.
Oh, yeah,
It's
okay.
just like a delay.
The
here.
big delay, okay.
Okay, so yeah, so it looks like that. And
Okay.
because I didn't do anything in the step functions, so
Mhm.
if you run that, it doesn't show.
I don't think you need.
Oh yeah,
think
it
that's
has
all.
no taxes. Okay. Oh, because I didn't adjust the contact manager.
Yeah,
Okay.
make them big, like 100, 100, the volume, the target volume. Otherwise, they I saw one disappearing. And 100, the other one too, just to make sure.
I'm
And
going to increase this a little bit as well.
I would leave it low because you want the more lambda volume you set, the lower they migrate.
Oh. Okay. Okay.
Yeah.
Um.
And then in contact, you
So you have to like adjust these parameters to change the pattern, right?
yeah, I would
So
say
you want
for
to-
now, yes, but I would say for now, just say front and back internal to 5 so that they are cohesive.
Okay.
Front and back to 5, yeah. I will just change that for now.
Oh, okay.
And let's see what happens. Okay. Are they more or less cohesive? No, they're still breaking. Are you assigning
I think
the clusters?
Yeah, I think maybe 5 is not enough.
Okay, let's do 1.
Yeah, because like you have to like be smaller than half of the damage between it.
Oh yeah,
and
true.
medium.
Yeah, you're right.
So now it's
Nice.
pretty
Okay.
tight.
And the red cell is the front. Okay, it's working. Oh, you had one cell breaking, one
Which
cell
one?
broke.
This one?
And also do periodic boundary conditions. But
Okay.
that's it. That's basically it. This is the model.
Mhm.
It's so simple,
Okay.
isn't it?
Yeah. Oh, okay.
What?
Problem.
What?
No, I think yeah, you're right. It's at least like simpler than the one that you demonstrated.
Yeah, I'll show you the code for that. The other one is the code is enormous.
Okay.
Periodic, yeah. So you go into C3DML, plots, replace,
Oh, okay. I almost forgot it, so-
yeah, and then okay. That's it. Save
Mm.
and run.
Oh yeah, like there are parts.
Yeah. We can have them hate each other a little bit, or have them hate. Let
We
me
could
think.
try the Q pattern first.
Try reducing even more the energy. Let's go to like 0.2. Yes, 0.2. Yeah. You have to stop and run.
That just make them like more active.
Yeah, but they are still breaking.
Yeah,
Interesting. Yeah. I could manage to get mine to not break. But this is fine. This is going well. Oh,
You
interesting.
can always put a link between the centers of the two cells to keep it from separating.
Yeah, you can do that too. And you can have a variety of behaviors, like you can have them migrate side by side. You can have them do swirls. You can have them.
How do you keep the fields within the individual cells from interfering with each other? Because that was always the problem with the original simulation. That if you have two cells that are next to each other, then the field doesn't have a gradient.
So the field is not in the entire cell, it's only in the front compartment. So you can have chemotaxis towards the back compartment.
Basically, the chemotaxis is at the line between the two compartments within the cell.
That one is blocked. This specific one is blocked.
Okay.
Yeah, there is a tag in, yeah.
um,
Let me check in my original simulation and what are the contact energies I used. So I have the internal is 0.5.
this one.
Yes. And all the other ones are 0 because they don't matter.
Oh,
Yeah, none of the others actually matter. You can
Yeah,
even
but-
delete them. Oh no, these ones you need. You don't need the ones with medium. You can delete all the medium ones. Yeah. Yeah, this is something you can also generally do for contact internal. You just delete all the medium energies. And then for contact, I have 5, 5, 5, 5, 4, 5.
5, 5, 5, 5, 4, 5.
Yeah.
Mm, mhm.
And that should do. Well, mine don't break, so I think that's. Oh, but there is also the volume.
If I were you, I would probably like
Oh, I do have links. I do have a link. Yeah. Okay, I thought I didn't. But yeah, I do have links. So
so between the front
yeah.
and back.
Yes, between the front and the back. Yes. But yours are still looking very rough.
But yeah, they're not going to stay as
Hmm.
black, so
Yeah, but they do look very rough somehow. Oh, I have neighbor order 7 in the contact. Both contact. Neighbor order 7. And the other one too. Yes. This makes a lot of like, yeah, you will see. There we go. Yeah, that's it. Yeah, you don't need links. And in this case, they migrate in a line.
why would they change them? Changing the label order here matters so much. Like,
You are considering a larger number of pixels to calculate the contact energy. So the energy is effectively larger. That's why my values are so low. They are 5. Actually, 5 is very large value if you have a neighbor order of 7. Well, these three are swirling together. Look, and the other thing it does is as you increase the neighbor order, you round the cells more. They get rounder.
mhm.
So that's why they look at very round like this. Yeah. If you reduce to 6 to 5 to 4, they will get like more rough and less round.
Yeah.
Yeah, basically the same thing.
So-
Try to get them to migrate side by side now instead of like that, side by side. Mm-hmm. Yeah, that's yeah, that's a good attempt. Nice. Now try to make them migrate in a line, on a line, like in a queue, you know, one after the other, chasing each other's tails.
So just like swap the contact and everything.
You can try. Yeah. I have another idea, but you can try that. Hmm.
Oh, it's stable anyway. Interesting. So you have to filter back.
Can you make them you succeeded, that's it. Now can you make them always swirl and never do anything else, just swirl all the time? Can you do that?
Always swirl.
Yes, always like swirl. Like even if you put two cells side by side, they always swirl, like chasing each other's tails.
And I can't change the diffusion stuff, right?
That's not a good idea.
Okay.
Yeah. I wouldn't change the diffusion stepables. Yeah.
So I wanted to cancel the diffusion towards chemotaxis toward the medium. That would work.
Oh yeah, that's the actual idea. So go to the chemotaxis plugin.
Oh, I thought they were like they're all
Yeah,
they're all
that's
the
the
same.
answer. Yeah, that's the answer. So
Oh.
that you come here and then you can set like towards medium to 1, -1, for example, very low value.
So stay zero.
Yeah, or 0. That will do. They will always swirl, always.
So here it should be like in a queue, but
They'll always swirl.
Mm.
Yeah, they can't do anything else basically.
we just start to curl
Yeah, I actually I never saw this. This is very interesting.
because it's so long.
Yeah, they are resisting a little bit.
Yeah, those are pretty compressed.
Yeah. Try to get your relax your contact energies a little bit. They're hating each other too much. Just set them to what we had before. 5 instead of 10. And yeah. And you can keep front and back to 3. That's fine. Yeah, just save and run. That should do the trick.
Swirl from the middle. Like oh,
Yeah.
this swirl is very weak.
And it's interesting that they still do migrate to a preferential direction despite not having any migration towards medium, but they should eventually collapse and start swirling.
You can check the substrate field too.
Yeah, that's the internal field.
Okay.
Can you exaggerate your lambda a little bit more, like 20 instead of 10? Yeah. There we go. Yeah. Nice. Yeah, one cell broke.
Oh, yeah.
Yeah.
But you can't call it as swirling because
Yeah, no, yeah. I managed to, oh, there we go. And you can see that there is like one cell is split into two compartments, and the
yeah.
back is here, the front is there.
Yeah.
But yeah, this is the swirl. Yeah. And actually this is experimentally observed. There is a paper on this.
And then like that's also like split.
No, 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 the cells don't split like this.
Oh,
Oh, I mean,
really?
you're not
What
talking
is the
about the
ratio?
experiment. Okay,
Okay.
yes.
Oh, so it's just going to swirl, but not very stable. Like,
Yeah, I thought it was going to be much more stable than this. I think it can be, because I remember being able to have them always swirl, but I think there is a different setting in the contact energy to play.
yeah, and I think it also depends on the numbers of the cell.
Yeah,
If
yeah.
you have like more cells here, then it also like would be more difficult to achieve this stabilized
Mm-hmm.
cell.
Yeah, this is
Um,
very nice. I I am like I find this one of the cutest simulations in CompuCell. It's so nice. And in the last few minutes, I don't know if do you want to explore something else with this simulation?
do you have any like else contents to introduce or go through today?
Yeah, but I mean it depends on what you prefer. Like I can talk the next topic in 5 minutes, so we can spend like 10 minutes doing something else with this.
Okay, why not? Like so,
Mm.
for example, like what are other situations or cases that we could explore here?
Um, you can have them divide.
Ah, okay.
That's a possible one. So if you want to have cluster division, you go to CC3D project. No Python.
Mhm.
Add steppable. Down. And cluster mitosis. Give it a name. Okay. Save.
So it's
Save the other one too. There are two
Oh,
files to
okay.
save.
Just automatically.
Yeah, let's just take a look at this this cluster mitosis thing there.
Different the volume.
For cluster in cluster, if cluster volume,
Oh, I didn't let it grow, so
yeah. For cell, if cluster volume, what is the current cluster volume is 100. So set that to 200 instead of 250. Sorry, 200 is the 400 then.
um,
Yeah, under that condition of the mitosis, scroll down.
okay.
So cluster volume 400. And uh, for compartment,
I think so I think I should write codes to let it grow, right?
yes.
Mm,
For cell and cell list, yeah, there you can say cell dot target volume plus equals.
just one.
Yeah, let's see what happens.
Plus
Let's run
two.
this. It should work, not flawlessly, but it should work.
Do you think it's growing?
No.
I don't think it's growing.
Okay. let's take a look at the code. And 1 pixel per Monte Carlo steps is big.
Yeah,
So
yeah, it's
yeah,
just
we
like a
should be
very
seeing.
significant.
Let's go back. So you have 4 cell in sub, so list cell dot target volume plus equals 1. What doesn't work? You should look. Scroll up. Oh, I see. I see. Yeah. No. You have in your XML,
Oh,
you
yeah,
have to remove the volume settings
yes,
and have those set in Python
okay.
Yeah. in the start function. There. In no, in the inside the loop, yeah. For cell.
Here? Or in here, I think it's all,
No, in the start function.
Oh,
It has to be just self. And then you give them a target and a lambda.
four, five. And then it's,
Yeah, there we go.
and it's
That's it.
very quickly. Oh, my
Yeah, very fast.
So let me just like slow
Yeah.
down a little bit.
In practice, like when I'm building the simulation and there is a lot of fine tuning to that cluster mitosis thing. But that's it. Yeah, it works. Some cells break when they split.
Yeah, you
So
see
the-
like this part is
yeah. Yeah.
to move like along the edges of the object because it's separate from its
hmm
front. Yeah, like this
Mhm.
part. And this you see like along it.
Yeah.
So and as this system gets bigger, it's slower. Why it's slow?
Because there are more pixel copy attempts happening.
Oh, so just the simulation gets slow.
Yes.
Okay, and I guess it could not grow anymore because
Yeah.
the pressure is too high, so it's
Yeah, no, there is no pressure inhibition of growth. So they are all the cells are growing still, the target volume, but they are like fighting each other for who wins and it stabilizes.
no, I think it's because the pressure is too high, so
But there is no pressure inhibited growth. I mean, but you are right that there is
It's-
no space to
it's
grow and
difficult
then
to copy
yeah.
pixel, right? To like to get more pixels to get volume growing.
Yeah. Yeah, you can see how it's a little bit flawed. Like there are 2 islands of
This one.
yes, of front compartments. So there is a lot of fine tuning that goes in this simulation. But you can see collective migration happening. Look at these cells going up, these cells going left. Yeah, eventually they get to a very high collectivity parameter. And also depends on the parameters, so depends on the contact energies, on the lambda chemotaxis. But it's I mean I love this model, so pretty. Yeah. I'm still disappointed that there is no like high quality experimental data on collective cell migration with spatial and temporal resolution necessary to compare 1-to-1 like this.
have to blame it on those wet lab workers.
Yeah. Always blaming on them. But otherwise, this could turn into a very nice paper. And finally, because these two exercises happened so quickly, maybe not so quickly, but we could do them in the time scale of the class. And this one you did basically alone. There is this final topic of Compartmental 3D, which I think no student should end the course without knowing this exists. But you don't need to use it necessarily, you just need to be aware that this exists. So you can build simulations in full Python. So you have like a Python script and you say import cc3d, and then you go through a list of commands and you can specify the simulation and run the simulation.
To me, like you need XML.
You don't need XML.
Okay.
It can be a full Python thing.
But still, like you're taking advantage of the C++ engine.
Yes, you still do. Yes, yes. So under the hood, you still have the C++ running everything, but you control all the simulation in Python, and you have a single script. So you can call your simulation using the command line command, Python, and then the name of your simulation.py, enter, and then it runs. So there are many upsides and many downsides. It's very easy to scan parameters because if you have a script, like if you can call your simulation doing running Python simulation, you can build a script that calls your simulation file and changes the parameters and run and distributes the results and do this automatically for you. We have parameter scan things in Compusal, but let's say, for example, you wanted to run a parameter scan where a certain parameter varies depending on the output of your previous simulation. Okay, then you cannot do with our traditional JSON file. Okay, suppose you are doing a parameter scan where you are increasing the size of the simulation, but you want to do like, oh, if the simulation is taking that long, I want to stop scanning this parameter and scan another one. If you want to do this kind of decision making automatic, it's much easier to do if you are in full Python. If you are optimizing your simulation, so for example, if you have if you are fitting your model to experimental data, it's also easier to do when you can call your simulation like that, because you can turn your simulation into a Python function. And then you call like a SciPy optimize, and then you use your simulation as the argument for the SciPy optimize. And then you find the best parameters of your Compusal simulation that fits the data in the full Python. Also, for example, in Monte Carlo algorithms, so that there are other things like many other there are so many Python functions that require your model to be a callable function. And when you have your simulation in full Python, it's usually easier. It's not that you cannot do with the traditional classic Compusal 3D, but it's easier if it's like that. Also, if you want to integrate with other Python libraries, for example, matplotlib, animation libraries, ray tracing libraries. So for example, if you want to make some cells transparent and look through the cells in a 3D thing, where they are like not completely opaque, but they are a little bit transparent, it's easier to have that if it's in full Python. Also, you gain all the benefits of your independent coding tool. For example, if you're using VS Code, you'll have all the AI assistance for coding built in. So you can use the AI assistance to develop your Compartmental 3D simulation, and to edit does not offer that. Also, if you have like neat plugins that you love in VS Code, you can use all of those to developing your. But you can also use you can also use VS Code and Player simultaneously. So you can develop your simulations using VS Code and then use Player to run them. So it's not that you can only do these things in a full Python simulation, but it's generally easier to do it that way. Among the downsides, so there is a steep learning curve. So it's very hard to get people to doing full Python stuff because the syntax is weirder. It's less human, not less human readable. I think it's maybe more human readable to my experience because XML introduces a lot of tags and stuff that don't contribute to the understanding. But it's harder to add it. You need Python experience. You need to set up it in a specific way. So you have to be careful because since you are editing VS Code, you have to select the Python interpreter. You have to link to your code environment. These kind of things can be troublesome. And the biggest problem, in my opinion, is that Player is very powerful. So you can do a lot of stuff in Player. You can select the fields here. You go to tools and then you generate a PIF automatically in tools there. You click, and then you have a PIF. You don't have, it's not as handy. You have to code everything when you are in full Python. So if you want to visualize the lattice, you have to code it. If you want to generate a PIF, you have to code it.
But just now you mentioned that we could generate code in like whole Python file, and we also use the player to visualize it. Or just I confused that.
No, you can edit your XML plus Python simulation in VS Code. But if your simulation is full Python, you don't enjoy Player.
Oh, okay.
Yeah.
Oh, I see.
Yeah, that I think, I mean, I never tried, but I don't think it's possible. If your simulation doesn't have, is not written like that with the three file split, Player will not, yeah, Player will not. And so this is one example of the kind of like, this is showing the upsides.
You're
and
not
downsides.
screen sharing.
Sorry.
Oh, no, I am not screen sharing. Okay, so this is the slide. So this is one example that this GIF shows upsides and downsides of it. So first upside, in this GIF, you can see that the cells are started in a circular configuration, but already in a hexagonal distribution. So if I were to zoom in, I don't know, yeah, I can zoom in. You can see that they are generated like quasi-hexagonally. So the waiting time to relax the simulation is basically 0. And how did I generate this initial condition? With a lot of AI assistance. I was coding myself and using the VS Code built-in AI tools to generate this. So it was very, very fast and easy. The other thing, this simulation is very big. And I'm running like thousands of these simulations because I integrated, I have a script that runs this in a cluster. So I can run this thousands of times and have thousands of GIFs. But the downside is already here. Like where are the cell borders? I don't see cell borders here. I don't have cell borders because I didn't want to code it. I would have to code the cell borders to visualize the cell borders, right?
There are like individual cell instead of like pixel.
Yeah, I'm basically coloring each cell a single color because I cannot draw the cell borders. This is something that only player has, and it's very hard to code yourself.
Even though like, they probably go to the same cell type, but you have to like.
Yes, they are all the same cell type. Yes. So there are many annoyances, especially with visualization, when you are doing full Python like this. But it makes your life easier. For example, right now I'm running these simulations in a cluster at the thousands, many replicates. And it's much easier if you can just say Python simulation.py, right? So yeah, that's it. That's it for today. If you have any questions, I can also show you how it looks
I have a
like.
question. What if your code is like there is some error in your code and how do you like retrieve the information, like error information from terminal or from terminal?
Yeah,
It will like report you.
yeah. And it's even better for finding your errors because it links you to the errors. So for example, if I put an error here and I save and run, it's going to give me a link to the error so I can click here and then it shows me the line. And if the error is in another file, it gives me the link to this other file. It's going to show here. And I can also ask the AI chat like, okay, what is the problem with this line? Help me understand this error message. And it has oversight over your project. So it's very easy. And the other thing you can do, and this is one I love, you click to, you come here and you say compare selected. And then it compares two simulation projects. So let's say you are versioning things or you are collaborating with someone and they made changes to your simulation. You just come here, like you have changes of your own, they have changes of their own. You come here, you compare them and you say, oh, this line isn't there. You come here and you say merge, and it's going to add this line there, vice versa. So you enjoy all the features of VS Code. The VS Code is extremely powerful. It has automatic linking. You can automate linking with GitHub. So all the changes you do here, you push to GitHub through VS Code. When you're editing your simulations into Edit, you don't have that.
Yeah.
So it's like a mountain of things to talk about in terms of pros and cons. But of course, like I run this like this simulation, and I don't see anything. Like it's running right now, and I don't see anything. So I have to code it myself. I have some lines here. If I uncomment one specific line, where is it? Here. Then I will see it. Yeah, this is the simulation. But again, like, what if I want to change the cell colors? What if I want to, switch to a field visualization? I don't have player. I'm stuck.
Right.
Yeah,
Also like a corresponding code to do that.
yeah, exactly. So it can be very limited in many aspects.
But you can also like target at one specific cell inside of it, right? Like the functions are similar to the tradition.
Yes, yes. The steppables look the same. If I like this is weird for you, like specs generation here, maybe not completely weird, like this is the contact plugin in Python. So it looks good. And you can also say, oh no, this is not n, this is np dot the square root of something. You know, you cannot do that in XML.
Yeah, well, we should change the XML so it parses the expression. It parses the value of the tags.
Yeah.
A
Then
better parser would do that.
yeah. Then here there is all more weird stuff. If name main, then you have a sim service thing that you so these things I don't necessarily understand everything that they are doing. I'm basically following a recipe. And then there is the run, the init, the start, the visualize. Then there is a while to iterate through the steps.
It's similar to my project, like it's a simulation.
Mhm.
It's like a class, and it has some method.
Mm, yeah.
Something like this one is for registering.
Yes. Yes, this
also-
should be like standard approach for other types of
Yeah,
simulations, yes.
It's actually
The steppables is go ahead.
So do you have a custom functions or methods in this workflow?
Yeah,
So you could just add like functions in this class, in the
Yeah.
same class. Okay.
yes. But the steppables look the same. You have the init, you have the start
Mhm.
for cell, it's the same syntax for everything. So it's quite easy to translate like your simulation from XML plus Python to full
Yeah.
Python if you need.
Yeah. I see.
And like yeah. Here let's see what we have. We have we don't have anything special here. Like you could use all these things in your own in to add it, fine. But for example, if I'm using matplotlib or like any animation tool, it's easier to use it in this. Yeah. Right now I don't know if you have used cluster before. Have you?
What custom?
Supercomputer
Oh,
with many nodes.
I used HPC to process my microbiome data because it's pretty large and it consumes a lot of the memory resources, so I have to use HPC.
Yeah. Here, let me show you what I have here. I'm connecting to a supercomputer. And this I have my simulation. This code is the same as the one I was showing in the VS Code. And then I have a script. The script calls the simulation, generates a batch file that, so this is the call right here, cc3d. This is the path to the file, and it says Python, and then the path to the file. So this is why it's very simple, because the path, the call to the simulation is so simple.
Yeah, so you pre-installed CC3D on HPC.
Yes.
or-
Yes, we install Compartmental 3D in the cluster. Yes. Then you can set, for example, how many jobs you want to give to the HPC. What if you hit the maximum? What do you do? You can define your own parameter scan. So here I'm scanning parameters. So I have two values for one parameter. I have geometric scanning for another parameter. I have two values for another one. I have my number of replicates. And if I come, let me see if they are running. Yeah, these are my jobs running right now. So some simulations are taking, this is 6 hours. And I'm running like, I don't know, like it's 100 simulations running simultaneously. And they are many hours each of them. So like in my own computer, this is insane, you know. And they have so many more. This is a waiting line.
Yeah, pending.
Yes. Yeah, pending, exactly.
Okay.
Yeah. So this is very powerful, and it's like it's less of a headache when you have your simulation in a full Python script, a single full Python script.
Yeah, it's because HPC probably would not allow you to use.
It doesn't allow you to use player. Yeah. Yeah, you have to kind of go around player, like work around player to run things in the HPC. And it's a little bit more complicated, not extremely complicated, because I've seen other people doing it, and it's not like, it's not a disgrace, but it's more clean and straightforward to do with a single Python script. Yeah, I hope that was useful. So keep in mind this exists. There is there is documentation and demos on the full Python stuff.
Thank you.
Right. I
So do you think you're going to be ready to present next week for your project? Because officially you need to do a little presentation of your project results.
Yeah, I guess.
We either do it next week or the week after. You have a choice.
Yeah.
I mean, next week is the last week of class.
Mhm.
And I'm sure we can find something to do next week if you want to have a regular class meeting. And then we could use the same time the following week, which is exam week, for you to do your presentation. I mean, historically, when we've had a lot of students, we've had the students do a dry run, a practice presentation next week, and then do it again the following week. So you get to try it twice. But obviously, we want to be flexible for adjust to what you want. So you tell me what works for you best.
Probably, um, actually, like all of my classes are like ends this week. So for next week, like I'll be very flexible next week.
Well, maybe I'm wrong. Isn't this week? Is this the last week of class?
No, I think
Next week is the last week of class.
yeah. Actually, according to the like campus calendar, like the week after next week will be the last week, but
Is that? Let me check. I want to make sure if I got it right. I thought it was. According to my calendar, this week, next week is regular class, and then the following week is exams. The 5th of May is exams.
yeah, it's
So in principle, next week is a regular class week.
okay. So do you have time like around April 29 or April 30?
You want to get after finish, do it after your, if you have things that are due next week, and then you want to do the presentation on the May 5th, that's okay. And I
No.
can certainly meet with you on the 29th or the 30th. That's no problem.
Okay, I think this presentation would be the largest thing. Like I didn't have like any other final projects for
Yeah.
next week.
So you prefer next week?
Yeah, but
Okay.
I'm a little bit afraid that next Tuesday will be like a little tight.
I'm sorry, I'm having a hard time hearing. So
Oh,
Pedro.
yeah, so I was wondering, like, do you both have time on maybe next Wednesday or next Thursday?
Instead of on Tuesday, you want to meet Wednesday instead of Tuesday
Or Thursday.
or Thursday?
Yeah.
Yeah.
Thursday would
Okay.
be best for me.
Okay, why don't we move the class from next week to Thursday then, to
Yeah.
the 30th?
Okay.
Okay.
And I would just give the presentation at that time.
Okay,
Yeah.
okay. So
Sounds
I've
good.
just changed the schedule for next week. Great. Thank you so for being so willing to try things out and being so patient and getting through so much. We've never actually gotten through all this material in a class before.
Thank you, and I hope you get better soon.
Okay,
how are
yeah,
you
well,
feeling today,
I'm sure
James?
I'll be fine next week, but I didn't want to come and cough on everybody today.
Are you
Okay,
feeling better today, James? So.
so it'll take a few days. It's really just a cold, but it's still
Mhm.
one of those things. Okay, well, thank you, Pedro, for doing that. I learned a lot from watching you do it too.
No problem.